Welcome dear students. I am Dr. Divakar Mehta, Assistant Professor from Carmel College of Arts, Science and Commerce, NUMEM. The program you are learning is Bachelor of Science first year, subject Botany, Semester 2, course code BOC 102, course title Biodiversity 2, which includes vascular plants. Title of the unit is Biometrics, Numerical Taxonomy and Cladistics. Module name Cluster Analysis, Phenograms and Cladograms. In the previous two modules, you have studied about different aspects of characters, variations, OTUs, character coding, and also you have learnt about character weighting. Broad outline of this module is, first we will see what is cluster analysis, then phenograms, cladograms and differences between phenogram and cladogram. At the end of this module, you will be able to recall the terms cluster analysis, phenogram and cladogram. You will be able to define term phenogram and cladogram, and also you will be able to differentiate between phenogram and cladogram. We have seen in the previous module that data is organized into OTU and OTU matrix, that is OTU into OTU. So it will become a exhaustive data which will be very difficult to interpret manually by us. And so for such data matrix we need computers, and even for computers we have to condense the data in a simpler way, and that will be helpful for the computer to analyze further. So cluster analysis is one such method in which OTUs are arranged in the order of a decreasing similarity. Cluster analysis is a class of techniques that are used to classify objects into relative groups called clusters. Objects in a given cluster tend to be similar to each other in some sense, and objects in different clusters tend to be dissimilar. That is, whatever pair of OTUs you are trying to compare, and several OTUs also we will be trying to compare. So the one which are closely related, they will show more and more similarity, and the one which are placed in a distance, they will be having more and more dissimilarity. In botany, cluster analysis is a method used to determine the similarity between operational taxonomic units. In this method, the OTUs are arranged in the order of a decreasing similarity and analyzed to construct cluster diagram or phenogram. So what we are trying to do is, we are trying to bring more similar operational taxonomic units into closer together, and the one which have more and more differences, they will be placed separate or they will be placed far. There are three different types of clustering: hierarchical clustering, non-hierarchical clustering and two-step procedure of clustering. In hierarchical clustering, what we do, we will try to develop a tree-like structure, or the hierarchical cluster analysis will result into a tree-like structure. You can see here the image. So in this you can see different operational taxonomic units. They are numbered 1, 2, 3, 4. They are arranged according to their similarity in the form of a tree. You will find 9 and 12, which has the maximum similarity. They are placed very closely. Whereas if you consider 5th and 4th, they have lot of differences. They are kept far away. So you will find the more the similarity, they will come close together, and more the difference, they will be placed far. Non-hierarchical cluster analysis. It does not involve the tree-like construction as in the hierarchical analysis. Instead, they assign objects into clusters. What we do here, there will be huge number of operational taxonomic units, and we have to decide how many clusters we want, and that data or that number we have to give the input in the computer, and computer will analyze and give out how many clusters or how many operational taxonomic units belongs to that particular clusters. With more the similarity, they will fall into one cluster, and like that you will find different clusters which will include more similar elements or the taxons. Phenograms. Graphical representation of the results of hierarchical procedure is called cluster diagram, phenogram or dendrogram. Starting with each object as a separate cluster, the phenogram shows how the clusters are combined at each step of the procedure until all contained in a single cluster. You can see in this diagram, you will find first there will be pair of operational taxonomic units which will have maximum similarity. They will be brought together. Then the next taxonomic unit will be added which will be very closely related to this. Like that you will find they will go on finding the most similar to dissimilar, and ultimately you will find such a diagram, a tree-like diagram, that is called phenogram. Cladograms. A cladogram in botany is a diagram used to represent a hypothetical relation between group of plants. It is used in phylogenetic studies to visualize how the groups are related and their most common ancestors. A cladogram is not an evolutionary tree because it does not show how ancestors are related to descendants. It will not show any kind of how they are closely associated with the ancestors. A cladogram uses lines that branch off in different directions, ending at a clade. A clade is a group of organisms with a last common ancestors. Just see this diagram. Here you can see at the base, that is the common ancestor, the most common ancestor for all the branches. Each branch you can consider it as a clade. So you will find Charophyta, liverworts, mosses, club mosses, ferns, all these, each branch or each clade will have a common ancestor. For example, you go to the top, that is gymnosperms and flowering plants. For gymnosperms, seed-bearing plants are the ancestors, common ancestors, and from this the gymnosperms and flowering plants have originated. For flowering plants, the ancestors had flowers, they also had double fertilization, they had endosperm and fruit, which is not seen in the gymnosperm. So at the top level we will find angiosperm clade. Now come to the differences between phenogram and cladogram. A phenogram is a diagrammatic representation of organism based on overall similarity, whereas cladogram is a diagrammatic representation of the hypothetical relationship among organisms. Phenogram attempts to classify organisms based on overall similarity, whereas the cladogram will try to attempt to give hypothetical relationship among the organisms. So there is a huge difference. Phenogram will give the actual fact, whereas the cladogram will try to give a hypothetical relationship between the organisms. Phenograms do not consider phylogeny or evolutionary relationship. Cladogram used to give phylogenetic relationship. Phenogram based on convenient observable characters. So you will see what are the morphological characters or observable characters, that only you will list, and accordingly you will do the or construct the phenogram. Whereas for cladogram, it is based upon all the available information. It will be morphology, physiology, genetics, biochemistry, ecology, all the field, whatever information is available, all the information will be considered to construct the cladogram. Phenogram emphasize on current relationship. What is the current relationship among the operational taxonomic units? Whereas the The cladogram will emphasize on ancestral relationship, how they are associated or what are the common ancestors for particular clade. So these are the differences between phenogram and cladogram. To sum up, in this module we have studied about cluster analysis, phenogram, cladogram, and the differences between phenogram and cladogram. These are the references. Thank you.